Okay, I'm going to talk about Ardipithecus ramidus and the evolution of hominin sociality. The data on Ardipithecus ramidus was published in 2009. It's believed to be an arboreal bipedal ancestor of modern humans. It has reduced canine morphology, reduced prognathism of the face. Now, I'm interested in applying Stephen Jay Gould's theory of heterochrony to A. ramidus. Gould published this book in the 1970s, and he was interested in the dissociation of developmental growth trajectories as sources of evolutionary innovation. And the model he used was salamanders who can retain larval morphology into the period of sexual maturity if they remain in an aquatic environment. And he looked at a similar phenomenon in humans and believed that humans retain juvenile craniofacial shape dimensions into the period of sexual maturation. So I'm interested in looking at Ardipithecus ramidus in chimpanzees and looking at what I believe may be synapomorphic traits and apomorphic traits. Ardipithecus ramidus shares a similar brain size with chimpanzees. Dimensions of the cranial vault are similar, but dimensions of the face are different. Now we can infer various life history trajectories, looking at correlations between brain size and the ontogeny, which I'll look at in a minute. So I think brain size and life history trajectories may have been synapomorphic in chimpanzees and A. ramidus and inherited from a common ancestor, whereas craniofacial dimensions I believe may be apomorphic, which means that Ardipithecus ramidus reduced craniofacial dimensions. Chimpanzees may have actually increased projection of the face, but this is hard to tell because we have no evidence of a common ancestral condition. So in order to find out the ontogeny of Ardipithecus ramidus, we looked at correlations between brain size and M1 eruption and brain size and age of first birth in the literature. And brain size provides a pretty good means by which to predict these other aspects of development. And as you can see here, A. ramidus clusters around chimpanzees and other great apes, different to lesser apes, different to humans. So we can assume that it possibly had a similar life history or ontogeny to chimpanzees and possibly bonobos. So I'm interested in looking at the dissociation of sexual maturation from the ontogeny of facial growth. In the data that White and Lovejoy and Sewer published, they looked at craniofacial projection in A. ramidus and compared it with adult chimpanzees and adult gorillas, and they found that there was a lack of sexual dimorphism and that A. ramidus had feminized craniofacial morphology. It clustered around the females of bonobos or paniscus. However, they did not do an ontogenetic analysis, so they were just comparing A. ramidus with adults. I was interested in comparing A. ramidus with infant and juvenile chimpanzees. In order to do that, we had to change the landmarks. Lovejoy and White and Sewer, they measured porion to nasion and porion to procyon to gauge degrees of facial projection. Because porion migrates backwards through great ape ontogeny as the cranial base opens up, we had to change those landmarks because porion is in a different place in an infant chimpanzee as it is in a adult chimpanzee. Interestingly, in Ardipithecus ramidus, it's in the similar position as an infant chimpanzee, that is, in the middle of the skull as opposed to moving backwards. So instead we used opisthocrinion instead of porion, and we measured nasion to nasal aperture as a percentage of the entire length of the skull from opisthocrinion to procyon, and nasal aperture to procyon as a percentage of the entire length of the skull from opisthocrinion to procyon. And we looked at the ratios there, and you can see Ardipithecus ramidus in black. The adult chimpanzees are in grey, and we have some juvenile and infant chimpanzees in the darker colours, and you can see that A. ramidus in terms of upper facial projection shows greater affinity with the juveniles than with the adult chimpanzees. And same again here, the adult chimpanzees are in grey, in light grey, and the juveniles and the infants are in the darker grey, and A. ramidus in the black, again much greater affinity with juvenile and infant shape dimensions than with adult shape dimensions. So what's going on here? If this is actually what's happening in A. ramidus, why do we find this lack of affinity with the adult great ape form. And I was interested in this paper by Cobb and Higgins, and they looked at the ontogeny of sexual dimorphism in facial skeleton of African apes. And they found that males and females tended to retain similar dimensions and a similar ontogenetic trajectory until puberty, and that's when the craniofacial sexual dimorphism seemed to kick in, and they suggested it was under hormonal control associated with puberty. And particularly, males develop a greater degree of subnasal projection at puberty than do females, and this accounts for the greater projection of the face in male chimpanzees. So why are males developing this greater projection of the face? And they said it's because, not so much because of the larger canines, but more because of the greater distance required between the upper canine and the incisors necessary for canine occlusion, and the CP3 honing complex where the canines, the upper canines, hone against the lower premolars, and they're sharpened for combat and tournament behaviour. So it was this diastema or gap between the incisors and the canine that produces this greater projection in the male face relative to females. A. ramidus has none of this CP3 complex, and the males and the females are thought to be very, very similar. They've not diverged at puberty as in other great apes. Consequently, how are we going to think about Ardipithecus ramidus reproductive strategies? In baboons and in other primates, these big canines are the means by which males vie for status within the sexual hierarchy, or predator defence or intergroup conflict. So they're used in conflict and threat display and these kinds of things. If A. ramidus doesn't have them, how was this species reproductively successful? And I suggest that the absence of canine armoury means that A. ramidus males must have adopted alternative reproductive strategies that did not require canine armoury. I was interested in Triver's idea of paternal investment in offspring, and he believed that the greater paternal investment there is in offspring, that will disproportionately decrease male energy expended in male-male conflict to inseminate females. So I'm thinking possibly that growing these big kind of projecting jaws and canines is metabolically expensive. So I'm thinking that prior to puberty, Ardipithecus ramidus diverted metabolic energy associated with craniofacial growth and conflict over females, or conflict with males over females, sexual access to females, diverted that metabolic energy into paternal investment in offspring. And this may have been provisioning of females or assisting in the raising of offspring, or but being part of a system of alloparental care or cooperative breeding. And we see this kind of thing in marmosets and tamarins, where if males invest in offspring, then that increases maternal reproductive output. So if the males help the females, then they can increase their litter size or reduce their interbirth interval. Now, I think that A. ramidus suggests that alloparenting, or some kind of involvement in maternal metabolic allocation to infants by other individuals apart from the mother, may have occurred much earlier than has previously thought. Now, generally people think that alloparenting developed with Homo erectus, because that's where we find the first evidence of increased body size and increased brain size. But just because these things exist in Homo erectus supposedly does not mean they evolved together. Alloparenting could have evolved much earlier and then been kind of the foundation upon which increases in brain and body size developed. Now I'm interested in looking at Robert Martin's maternal energy hypotheses. Now, he believed that the increase in brain size that we see in the hominin lineage was facilitated by an intensification of parental care, and he was interested in maternal metabolic allocation to infant growth, to fetal and infant growth, sorry. Martin followed Adolf Portman's notion that humans are secondarily altricial. We're born very immature. We have a greater period of infant dependency, and he was interested in the notion that humans tend to extend in utero brain growth rates ex utero, and following Portman, Martin believed that human gestation is actually 21 months, in that in utero growth rates are extended ex utero an extra 11 months, and that accounts for the fact that we're born very, very immature. Now, I'm also interested in the role of females and women in human evolution, and particularly the work of Adrienne Zillman. She developed the Women the Gatherer thesis in the 1970s in response to the Man the Hunter thesis, which proposed hunting on the part of males, and the provisioning of females and their kind of helpless infants was what led to the process of hominisation and our divergence from the apes. Whereas Zillman wanted to emphasise that really what was going on was an intensification of maternal care and a change in maternal patterns of socialisation of infants. Now, she proposed that if there was a greater period of maternal socialisation of offspring, this could produce pro-social behaviours in males, and if those behaviours were carried into adulthood, that they could differentially proliferate through female mate preference. Now, we find this in baboons. Barbara Smuts did work on baboons and female mate choice, and as opposed to earlier models which thought baboon society was structured around male dominance hierarchies, she highlighted the degree to which female mate choice was very, very important, and even in in very sexually dimorphic species as baboons. And Zillman argued that reduced canine morphology is evidence of female selection for less aggressive males, so again reinstalling the role of female choice within human evolution. And changes in social and mating systems precede archaeological evidence for technological and cultural innovation. So she believed we diverged and changed our cultural and mating systems millions of years before you have evidence of hunting and cultural and technological innovations in the genus Homo. And there's been quite a lot of work written about this. Zillman had a debate with Lovejoy for the last 20 or 30 years, she debated Lovejoy. Lovejoy carried on as though Zillman didn't exist. But Lovejoy proposed in Ardipithecus ramidus that bipedalism is evidence of male provisioning of females. So you have these kind of sedentary females who are kind of gathered around the fruit tree, and the males go off and provide food for the females with their more dependent offspring. And now they've got dependent offspring, they sort of can't move around and they're burdened by these late maturing offspring. Zillman said she couldn't find a hunter-gatherer community where this was the case, nor is it the case in chimpanzees. It was the case in 1950s America, where men go out and bring back, provision kind of sedentary females. But she looked at the Kung and found that the women actually carry the infants while they're foraging. So infants do not prevent them foraging, and the Bushmen the Kung Bushmen women provide 70 to 80% of the food. So she questioned the whole male bias of Lovejoy's theory and various other theories that looked at a kind of male emphasis in human evolution, reinstalling the role of women. So Zillman believed that carrying of infants facilitated was necessary to kind of the development of altriciality, that you could forage for food and carry infants if you were bipedal. So it's important for female or maternal care. I'm also interested in oxytocin in humans, which is a mammalian nurturant neurochemical, and it establishes the mother-infant bond. But also, interestingly, in humans, oxytocin not only is involved in the mother-infant bond or maternal care, it's also involved in male sexual psychology. So during sex, oxytocin is secreted in the male brain, and it's thought that this may establish deep emotional bonds between males and females that facilitate this shared division of labour or pair bonding that underpins our unique mating systems and sexual psychology. Conclusions. Juvenilized craniofacial morphology in Ardipithecus ramidus. It's a feminized morphology, and possibly male social emotions have become feminized as well, in tandem with the feminization of the facial morphology. This means there's a divergent ontogeny at the base of the hominin clade, so our divergence from apes is very, very early. A dissociation of craniofacial growth from sexual maturation suggests change in mating and social systems. Primacy of maternal care in the process of hominization. Changes in social and parenting systems, probably the basis for later innovations such as cultural and technological innovations. A chimpanzee referential model is probably no longer applicable because we had a different fundamental ontogeny at the very, very divergence from the common ancestral condition. So models such as Richard Wrangham and Craig Stanford that use chimpanzees to reconstruct early hominin behaviour, I don't think are really are no longer tenable. The origins of hominin sociality in late Miocene and early Pliocene forest and woodland ecologies as opposed to savanna ecologies.